Yeah, so on behalf of the co-investigators, I presented the dose escalation component of the phase 1 clinical trial of BGB-3245 in patients with advanced solid tumors. As some background, the MAP kinase pathway is among the most commonly altered pathways in cancer. BRAF mutations can be classified into 3 different groups on the basis of how they signal. Class 1 BRAF mutations signal as monomers. This is typified by BRAF V600E and accounts for greater than 90% of BRAF alterations in cancer. And they have high kinase activity, signal as monomers, and feedback suppress RAS. Non-V600E mutations can be subclassified into class 2 or class 3. Class 2 mutations have high kinase activity or intermediate kinase activity and signal as constitutively active dimers and feedback suppress RAS. Class 3 alterations have low kinase activity or are kinase dead, and they signal as RAS-dependent heterodimers. So first-generation BRAF inhibitors, including Vemurafenib, Dabrafenib, and Encorafenib, have been approved by regulatory agencies for the treatment of BRAF V600 mutant cancers, so class 1 alterations. However, they have several limitations. So they do not effectively treat class 2 or class 3 alterations, BRAF fusions, splice isoforms, or N-terminal deletions that signal as dimers. Additionally, they're ineffective in RAS-mutated tumors. Also, they cause paradoxical activation in wild-type cells, and that leads to keratoacanthomas and cutaneous squamous cell carcinomas. Furthermore, patients with BRAF V600E mutant cancers that are treated with first-generation inhibitors can develop acquired resistance, and that can be mediated by dimer-mediated mechanisms that could potentially be amenable to different types of therapies. So BGB-3245 is the drug that we discussed, and that is a BRAF dimer inhibitor that inhibits all RAF isoforms with strong potency. It is in preclinical models effective in class 1, 2, and 3 alterations, and has the potential to be effective in BRAF fusions, splice isoforms, and N-terminal deletions. This drug should not cause paradoxical pathway activation at therapeutically relevant exposures. So we discussed the dose escalation trial. This was a phase 1 dose escalation that followed a modified toxicity probability interval 2 design, starting with a dose level of 5 milligrams once daily. The primary endpoints were safety and tolerability, the determination of the maximum tolerated dose and the recommended phase 2 dose, and secondary endpoints included anti-tumor activity and pharmacokinetics. 6 dose levels were explored, and we identified 40 milligrams once daily as the maximum tolerated dose. In terms of the toxicity, so the toxicity profile was consistent with that of other MAP kinase inhibitors. The most common side effects included rash, fever, thrombocytopenia, and ALT elevation. Generally, these were low-grade and manageable. 12% of patients required a dose reduction. In terms of the efficacy, I should say the patient population. So in the patient population, we described 42 patients as of the data cut, including 9 that were ongoing. These patients were heavily pretreated with a prior median line of 3 prior systemic therapies, and many different disease types were included, most commonly melanoma, non-small cell lung cancer, and colorectal cancer, although there were many different diseases. We treated RAS mutations, KRAS, NRAS, and HRAS mutations, and BRAF alterations, most commonly BRAF V600E. However, class 2 alterations and BRAF fusions were included as well. Overall, there was a response rate of a confirmed overall response rate of 18%, with an additional 24% of patients having stable disease beyond 24 weeks. The disease control rate was 79%. Confirmed responses were seen in a variety of different tumor types and genomic alterations, including BRAF V600E mutant cancers that had progressed on prior BRAF and MEK inhibitors in melanoma, cholangiocarcinoma, and in low-grade serous ovarian cancer. Patients with NRAS mutant melanoma had confirmed responses, and patients with BRAF class 2 alterations as well. The median time on treatment was 154 days, and many patients continued treatment beyond a year. So In summary, the dose escalation of BGB-3245 identified 40 milligrams once daily as the maximum tolerated dose. In general, the safety was tolerable and consistent with other MAP kinase inhibitors, and there was preliminary signs of efficacy observed in several different populations. Importantly, many populations for which there is no currently approved targeted therapy, and there is significant unmet need.